Bismillahirrahmanirrahim. After the introduction of the enzyme, we will study about the cofactor of the enzymes. Cofactors are the non-protein part of the enzymes which are required for the action of enzymes. If there is no cofactor attached with the enzyme, there will be no activity of enzyme. Some enzyme, not all the enzyme, some category of enzymes required a non-protein part, that sometimes it organic, sometimes it is inorganic. So different cofactors are needed for the specific enzymes, and these enzyme when attach that cofactor, then the enzyme become complete. Without cofactor, like this is an enzyme and it is without cofactor, the cofactor is not attached with it, that will be known as apoenzyme. Without cofactor attached, the protein is not active. It will be active only when the cofactor is attached with it. And when the cofactor is attached, the enzyme at that time is known as holoenzyme. Without cofactor, it is apoenzyme, and with the cofactor, it is holoenzyme. And when the cofactor attach with this enzyme, then active sites arise, or amino acid adopt itself in a shape of the substrate, matching with the substrate. Then the substrate attach with the active site of the enzymes. We can study in your book, the example of the pepsinogen is given, that it is an enzyme which is responsible for the digestion of the protein present in the stomach. Pepsinogen is an apoenzyme because that is without cofactor. And when cofactor attach with the pepsinogen, it become pepsin, holoenzyme, and it can actively work on the protein only in the form of pepsin. Cofactor have different types. Like there are the three types of the cofactors that are responsible for the activity of the enzymes. Number one, it is the activators. Activators are the inorganic molecules that attach for the enzyme activity. Like here, the iron, magnesium, copper, zinc, etc. These are the inorganic ions or metallic ions which are responsible for the activity of enzyme. You can see here that the reactants are the substrate, glucose and ATPs can change only to glucose, 6-phosphate and ADP. This is the reaction, the whole reaction, and here the enzyme hexokinase is working, but it needed here the ion magnesium for its activity, and this magnesium act as an activator. And these are detachable. Detachable means that it can attach to the enzyme only at the time of reaction, and after the completion of the reaction they detach or separate from the enzymes. Another cofactor is the organic molecules, and organic cofactors can be divided into two categories, that is the coenzyme or prosthetic group. Coenzyme are the organic compounds like sometimes ATP, NAD, FAD. These work with the close association with the enzyme. They attach with the enzyme only at the time of reaction and then can be detached like the inorganic. They helps the enzyme in their work for specific time and then detach or separate from that enzyme. While the prosthetic group is a group of organic compounds, organic molecules that attach with enzyme, but their attachment is permanent, means they are covalently bonded with the enzyme and they don't attach from the enzyme during their need during their chemical reaction, but they are the permanent parts of the enzyme. They are the permanent member of the enzyme, structural part of the enzyme that attach with them anytime, and they don't detach from them after the activity. Now, the mechanism of enzyme action, that how enzyme act. Enzyme normally form a complex with the substrate. When enzyme combine with the substrate, it form a complex which is known as enzyme substrate complex. After the formation of the complex, the active site of the enzyme having the catalytic site become active, and it change the substrate into product. So the complex now change from enzyme substrate complex to enzyme product complex. After the formation of the product is then detached. The bonding of the product with the amino acid of the enzyme is broken down, and the product is changed, the product is separate, detached, and then enzyme again active for the reaction of another substrate. So enzyme work in this manner, enzyme act in this manner. The two type of the models are proposed by different scientists for the reaction or for the action of enzyme, in which one is log and key model and the other is induced fit model. This is the model which show the log and key model. Log and key model is proposed by Emil Fischer in 1894. And according to this model, he said that the active site of the enzyme work as a log, and the specific substrate which attach with the enzyme work as a key. So as the specific key can only open a specific log, like that the shape of the log and the key must be matched, and when it matched, it combine with each other and change into product, but having no structural changes will be occur in the enzyme. Our enzymes are not flexible. They are not flexible. There is no change in the structure of enzyme with the attachment of the substrate or the formation of the product. So this model is known as log and key model. Like there is no change occur in the structure of the log when we insert key in that, just it open the log. So like that the substrate attach with the enzyme and it change into product, and having no flexibility or structure changes occur in the enzyme. And those enzymes which follow this type of model is known as non-regulatory enzymes. And this is the model showing the induced fit model proposed by Koshland in 1959. According to this model, he said that enzymes are specific to their substrate like log and key, but they are flexible. There is flexibility in the structure of the enzyme. When enzyme attach a substrate, it causes the changes of the structure of enzymes, and these changes are flexibility work the enzymes for more than one substrate. Like when the changes occur in the structure of enzyme, it carry out its substrate into product, and then the enzyme after the detachment of the product, it form its original shape, and then it convert another substrate of the even of the same category. Like as already we have discussed that enzyme carbonic anhydrase, it not only combine oxygen with the hemoglobin, but it also responsible for the formation of the carbonic acid and bicarbonate in blood. So like these enzymes which work for more than one substrate, they are flexible. When the substrate attach with them, they causes changes in their shape so that the catalytic side become active, and when catalytic side active, the substrate change into product, and then the product, then when detached from these enzyme, the enzyme regain its original structure, and this model is known as induced fit model. And the enzymes are the material which the enzymes which follow this type of model are known as regulatory or allosteric enzymes.